Good old square pusher. I'm not a big fan of his newer stuff, though. It is a little bit of feedback there. It is 5:06 p.m. afternoon, Monday. Monday, April 21st, 2025. Mark Kulak, Talk.live. And my energy's been a little bit low lately. Apologize for that. I'll try to rally a little bit for today. Certainly no lack of content. Hope you guys are well. It is April 21st, 2025, 5:07 in the afternoon. Mark Kulak, Talk.live. I think I already said that, but whatever. So good, might as well just say it again, right? And we are halfway through the Judith Miller Germs book here because we are hardcore. We're not into just throwing every single potential wacky, stupid conspiracy against the wall and then self-congratulating ourselves for it. We are doing the books which actually matter, the books which are written by the mainstream sources, by the people who are as inside as you can imagine, not some, Johnny-come-lately authors who pop up after the fact and are just throwing conspiracies out there, which no one really cares about, to be honest with you, and could never even be repeated in a court of law. But this stuff is the real deal because these are working with the real people, such as Germs. So many people that were involved in the first Gulf War, in the foreshadowing of a potential biological terrorism attack in a major U.S. metropolitan center throughout the 1990s, and even the response to September 11th, etc., all the way up to the present. These are the books that matter, and they ain't easy books by any stretch of the imagination. As I said in a video I did last week, a shorter video that I didn't share yet. Chapter 5, I need to redo the narration for that a little bit. I'm not sure I'm going to invest the time in redoing all of it. My inflection is just not that great. Again, when taking the Amazon audiobook for Germs and putting it into nice chapters that align with the chapters in the book, when I did that for chapter 5, at that time, when I did that audio, I didn't know how much of it was going to be missing. So I read a sentence or two into the microphone on my laptop while I was cleaning it up. And yeah, the tone and the inflection wasn't great, but whatever. I was just filling in a sentence. And then I realized I needed to add a paragraph. Then, oh my goodness, I realized there was a bunch of paragraphs missing at the end. But I didn't put the effort into it to redo it perfectly. And then eventually I realized, oh my goodness, 30% of that chapter isn't in the audiobook. And then I went back and did it properly for the chapters up into that. But I didn't redo chapter 5, so it's a tough chapter to listen to, and I apologize for that. This book, Germs, and we need to get back to this, and we need to finish. We need to complete this book, as painful as it is. And you know what? If there's only 3 people that listen to me, fine. I'm going to do it right. I'm going to finish this book. We're going to listen to the audio. And then I may actually need to listen to it again several times, at least twice, because there's a lot of stuff in here that's missing. One of the first big things that's missing is major observations. First of all, I didn't know how liberally Joshua Lederberg could be referred to going up to September 11th, 2001, and how he was, well, you could still find newspaper references to him afterwards, but boy, his life was very different after 9/11, for sure. I think I think he lost favor with some people. Let's just put it that way. Maybe some people that you don't mind losing favor to, if I might be direct about it. There was a mention made to anthrax letters today on the This Week in Virology show, the kind that make you sick. Vincent Raccanello, Rancid Yellow, and his frequent bow tie doctor guest were made a mention of that. And something else I found to be quite curious, and I'm going to follow up on that towards the end of this particular program here today. I'm going to follow up on that because I find it interesting how these people who get hundreds, thousands, tens of thousands, hundreds of thousands of views, I don't know, who are so visible, influential, maybe, are so close, are basically dancing around the stuff that I've been covering on this program. And maybe they don't know, but I think they know. I don't think Vincent is a dummy. I don't think he's a dimwit. Maybe he doesn't know everything. But there's no way, even if you're not aware of the full spectrum of what's going on, okay, you have to be reasonably high functioning to get to that level. You can't hang out in the laboratory of Dr. Baltimore for years and then get your own program and be trusted to maintain a narrative if you're just not very sharp. All right. So Vincent has to be at least modestly capable. Not that he's a brainiac by everyday standards, but he has to be capable. He has to know some of this stuff. So I'm going to cover that in a minute. The first thing I wanted to the first part of the program, though, is I wanted to just highlight and I'm not going to go into it 100% because I really want it to be, I really want everyone, including myself, I want to enjoy it. Jonathan Cooey's show, and as he goes through this biology 101 for all of us as he presents it, and perhaps because I get into the history stuff, and it's interesting, right? I'm able to get into historical connections and mentorship connections and patent issues and so on, and that sort of has helped direct him as to areas of of biology which have been perhaps misinterpreted or are relevant but have been hidden, if you will. And likewise, sometimes I understand areas which matter more from the mentorship chains, corporations, because of the biology that he talks about. And the further I drift into if I make the mistake of pretending to be a molecular biologist, it's pointless. There's no reason to. So I have to really stay within my lane there. But I'm still capable of reading papers. I don't have a laboratory, but I am capable of reading the papers. And the things I wanted to highlight that he has been evangelizing, one is this word phage, P-H-A-G-E. And if you wind up texting it, it's going to be autocorrected to page all the time. So it is what it is. Phage, P-H-A-G-E, or bacteriophage. A bacteriophage is a virus that is unique or specific to the whole animal kingdom, whatever you want to call it, life kingdom of bacteria. It's pretty simple. Bacteria have viruses, multi-cell, eukaryotes have viruses. Prokaryotes have viruses, eukaryotes have viruses. And they're different. Okay, they're specific to the kingdom type. I mean, it's pretty simple. And we've observed that there's oftentimes bacteriophage is simply called, or phages are simply called viruses. And it's been said, there's trillions and trillions of viruses in the human body. Well, yeah, most of them are actually phages. The majority of virus biological entities are phages that are in the human body. It's probably the most abundant biological entity or unit within the human body is a phage or a bacteriophage or a virus that's specific to bacteria. And they're not there just to kill bacteria and annoy bacteria. They're there as part of the mechanisms that bacteria use to communicate with each other and to maintain their own symbiotic relationship. And they're also used in ways which we're just starting to get a handle on it, to actually to even manage the symbiotic relationship between cells and the bacterial kingdoms or colonies that live within a human body and make life possible for mammals. No one will ever call a virus for a multicelled organism a phage. For some reason, that mistake is never made. However, oftentimes you'll hear, oh, there's just tons of viruses, which include phages. I don't know why. It's a little trick that I've noticed that happens a lot, and it's kind of a disingenuous thing. If someone wants to use the word virus and to say this refers to these entities that aren't really living and are used for eukaryotes and prokaryotes, bacterial and multicell organisms, then fine, and they should just say it's viruses of both types. But they don't. So there's a lot of misleading language use. And it's clear that, as Jonathan well articulated in his last show, that no one wants to mention phages. Nobody wants to mention phages. They just don't exist. They'll argue about isolation of viruses. And then the whole world of bacteriophages is just ignored. It's the largest signal that you could actually find out there. It's the most abundant biological entity, as far as quantity goes, in the human body by a wide margin. So why are they not being spoken about? And they're just not there because. They're there for a reason. They're doing stuff. Not necessarily bad stuff either. They are doing things. They are being used to make us live. Those 200 trillion, approximately, give or take a couple, bacteriophages that are in a human body are there to help us work. Why won't anyone mention it? And why won't the virus deniers ever mention it? They don't mention it at all. Just they don't exist. So, and the people who are on the team of either worst case scenario or talking about lab leaks and all that stuff, they'll never mention it either. It's a word that's never used, and it's a critical word. And we're not saying it's a word to use suggesting that everything about virology is phages. That's not what I'm saying. I mean, there's an abundant amount of literature. I don't need to be a molecular biologist. The literature talks about how they've observed cells of multicelled organisms budding off and making their own little packages where they are actually sending out genetic information, which is then received by other cells and so on. So, and that's exosomal communication. So that stuff is real too. So this is an important element, or I should say concept. Now, I also shared a video. If you go to my Rumble page here, let me add that input here. Okay, and this is again, I don't know why this is happening. I wonder if I need to do an update on these puppies here. Okay, well, I don't know why. That's working now. Okay, so this is my Rumble account, and oh, great. I shared this video. It's a 19. Look, and now it's not updating again. Gosh darn it. Sorry about this, guys. I'll just make something up along the way here. Add that in. There we go. It's a 1972 video titled Ecology of a Hot Spring, Life at High Temperatures, Thomas Brock Biology, PCR, Archaea. All right, so there's a little bit of a mouthful there. So you can see this cool-looking guy in a cap. It's a 1972 video that was actually made by the Encyclopedia Britannica film, and it's part of a longer series. It's a 12 or 14-minute clip. Now, in the 1960s, this scientist, Dr. Thomas Brock, B-R-O-C-K, and he passed a couple of years ago at the old age of 94, lived a good full life. Total American Wisconsin boy. If you saw any videos of him just talking, he looks like someone that would work at a gas station. He looks pretty cool, but he doesn't look like some scientist who's just totally full of himself. As a matter of fact, he was probably asked to dress up a little bit for this video here because he's kind of styling a little bit more. It looks like he's ready to break out a saxophone right there and try to romance a woman or something. He doesn't look like he's ready to be to collect bacterial samples in Yellowstone Park. But in the 1960s, he went to Yellowstone National Park in the United States, where they have these hot springs and this environment, which is both very sulfuric and acidic, high temperatures, and it's an environment which would typically not consider to be conducive for life. And he went out and he went to see if he could find any type of microorganisms living there. And sure enough, he found tons, a whole array of organisms living there, multi-cell organisms or single-cell organisms, which were initially called extremophiles, which sounds like something you would read in a Jeffrey Epstein book. But it meant to refer to single-cell organisms that live in extreme situations. Now, as it turns out, those extreme situations may have actually been, if one goes down the evolutionary path, the first organisms that ever lived. Now, those samples were kept, and as time went on, those samples were kept. They were put into libraries, they were stored, and they were ultimately isolated further. And one of the enzymes that came out of those experiments in the 1980s was used by a Dr. Kary Mullis to make PCR possible. Kary Mullis wouldn't have been able to create PCR if it wasn't for a very specific type of enzyme, which was found used by this bacteria at a high temperature, because they needed an enzyme for PCR that could survive certain temperatures. So it's not as if, not to say that Thomas Brock himself fully discovered PCR technology, but it wouldn't exist. Kary Mullis wouldn't have been able to make PCR if it wasn't for that particular enzyme, which was found by Thomas Brock here at the Yellowstone. I've heard about this story a year or two ago, and it didn't really occur to me what I was learning. It's like, okay, so someone found an interesting bacteria with an interesting enzyme, etc. And I just kind of, I filed it away somewhere in my brain, and I made a page for Thomas Brock, and that was the end of it. All right, and that's the sound that someone just came or went. Now, a couple things here. One is that Thomas Brock, he did a lot of work at, I think, Indiana, but he eventually did a long tenure at the University of Wisconsin. So you know that although he joined University of Wisconsin after Joshua Lederberg left, you can imagine that there was Dr. Joshua Lederberg had some sort of collaborative relationship at some point or another with Thomas Brock, and they definitely communicated moderately frequently. Yes, I need to get some things out of the garage when I'm done. And I find that to be noteworthy because Joshua Lederberg, who was of CETUS, who wound up becoming a scientific director of CETUS Corporation, CETUS, is the scientist who found a lowly, Beach Boy-looking Kary Mullis in 1983 at an exhibit, an internal exhibit for CETUS Corporation, where Kary Mullis had his poster board out, and this is the legend, right? Kary Mullis was saying, "Hey, here's my PCR technology," and everyone just kept walking by and thinking that this guy doesn't know what he's doing. But good old Joshua Lederberg went up to Kary Mullis and said, "Hey, I think this is going to be a useful thing." Well, actually, he probably would have said it in a much lower voice because he had a very distinctive way of talking. Joshua Lederberg did. So both Lederberg and Brock played a key role in Kary Mullis and PCR, and Gary Mullis eventually winning the Nobel Prize. Now, I'm not saying that Gary Mullis was a bad scientist. I'm sure he was actually a very good, perhaps better than average scientist. But it's very difficult to get that opportunity to have so many things come together for one person. And I think he might have been nudged a little bit. This is Dr. Gary Mullis. And again, Gary Mullis created that technology when he was with CETUS, where the scientific director was Joshua Lederberg, who we talk about extensively. Joshua Lederberg, oddly enough, is one of the main characters in our Germs book, September 11th, 2001. Okay. Now, so I would recommend, there's not many videos out there on Thomas Brock. If you go searching on YouTube, you're not going to find much. Very little found out there on Thomas Brock. He wrote a lot of books. He had like 300 papers, and the one of the first big books on, I think it was, let me see the title of it. One of the first, there's a massive textbook, and it is called, and I can search in my eBay history because it will be here, titled The Biology of Microorganisms, which is one of the more important books in college texts right now. The Biology of Microorganisms was written by Thomas Brock, and it's up to its 14th edition. It's still in print. It might be 15th or 16th edition right now. And the very first edition of The Biology of Microorganisms, and you can imagine this is a key college textbook, was written by Thomas Brock in 1970. And it just so happened that yours truly found the original first edition for $42 at a museum that was selling it on eBay. So I got both the first, the second, and the fourth edition all coming to me to be scanned in. Now, what Thomas Brock discovered with these particular, these unique and exciting bacteria that were able to live in these extreme conditions in Yellowstone Park. You could find similar ones in other places, such as in underwater areas where there's underwater or deep sea volcanic activity, places where there's heat, where there's unusual circumstances. You can find these types of bacteria, and as it went on, eventually this type of bacteria wound up falling into its own class of microorganism called archaea, Archaea. Now, typically, I'm not really into the whole, we need to know all about the different families of bacteria because it's way out of my pay grade. I need a microbiologist. I need a Jonathan Cooey or someone with a PhD to talk that language for us. But I think the word archaea is actually so important that I would encourage you to watch a few videos on YouTube and get familiar with it. Okay, Archaea. Let's see here. Matter of fact, let's just go to YouTube and let's search for Archaea. Matter of fact, I may just play one right now. And you'll notice a lot of videos lately. A lot of videos. Suddenly, everyone's been talking about this lately. This is a hot word, archaea, because when they first found these things, when this Thomas Brock first found this stuff, they thought it was a unique single-celled organism, and at first they thought it was a bacteria. But then they said because of its membrane and various other properties, it was actually borderline mammal. They actually put it into the eukaryote kingdom. But now there's debate about which kingdom it falls to, and they're actually creating a third kingdom of life called archaea. Did you know that? Because I sure as hell didn't know it, a week or two ago. I never even heard of this word, but this is a hot word. And it matters here because this archaea, first off, it was able to, they were able to harvest this enzyme from it that made PCR possible. If not for this archaea animal kingdom, there's no PCR, period. Go home. It's all over. And this and this cool-looking dude, ready to, play in the jazz club, Thomas Brock. I don't think, and he didn't dress like this every day. It's kind of funny. He found this stuff. This is what made PCR possible. Kerry Mullis didn't go out into Yellowstone to find these unique organisms. Thomas Brock did in the 1960s, and it's recaptured here in this 1972 documentary, this 12-minute documentary, Encyclopedia Britannica film. It's a little bit slow, but I think you'll definitely enjoy it. So there's a debate right now as to if this archaea is a single-cell organism or if it's a bacteria, a prokaryote, or a eukaryote. A very hot debate. And now there's even suggestions that it's its very own animal kingdom, the third kingdom, and it might even be, as far as evolution goes, it might even be the original form of life. Bacteria might have actually evolved from it, and this is really interesting. Eukaryotes might have also evolved from it. It might actually be the common ancestor of all life. Now, I'm a Christian, so I don't even know how to best quantify stuff. I don't doubt evolution, but I think I believe that if the story of evolution is true, that it's how when the dust or whatever you want to call it that created all of existence. Eventually, one thing led to the next and led to galaxies and solar systems and planets and then life, etc. That's just the way things wound up falling into place. So, for me, it's an interesting conversation, but I don't get too caught up in it. But many people do love these conversations about evolution, and if they are, you should be interested in this conversation of archaea. So this is one video. This is a 7-minute video here, and doesn't have a ton. 353,000 views. Oh, by the way, you can. Spoiler alert. You have archaea in your body as well. We all do. Matter of fact, we have archaea in our microbiome, in our gut. It's one of the most important biological entities living in our gut is archaea. And, by the way, they also make viruses. They also have ways of communicating. And those ways of communicating are actually closer to mammals, as far as structure goes, than bacteria. You might even notice, if you start digging in, that the viruses of archaea, you will find ones that look identical to SARS-CoV-2. Interesting. No one wants to mention phages. But let's watch this video here to get things kicked off, because this person here is better spoken on the subject than I am.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about archaebacteria, or archaea. Basically, when we first discovered archaebacteria and separated that as its own domain, we thought they lived just in the harsh environments on our planet. So we discovered them in the hot pots of Yellowstone Park, we discovered them in the Great Salt Lake, we discovered them in swamps. And so we thought these archaebacteria kind of lived on the edges of life, but we really hadn't looked everywhere. And once we started looking out here in the ocean, used a method called PCR, and that's polymerase chain reaction. It's basically a way to take a little bit of genetic material, so a little stretch of DNA, and then make millions and millions and millions of copies of it, and so we could actually study it. And what we found is that when we started looking in the ocean, almost 10% of all the life we were finding are archaebacteria. And so archaebacteria...
Whoa, first of all, right there. Now, I don't fault anyone for this, okay? But recently it has been discovered that 10% of all of this microbial life that they in the oceans was something that they didn't know was there. I want you to think that right now the understanding of what the background signal is, okay, as Jonathan Cooey says, if no one's been looking for white Hondas, and suddenly they start looking for them and they start finding them, that doesn't mean that there were no white Hondas the day before and the day before that. These things have been there. They didn't just appear. They didn't even know they were there.
They are everywhere. They're found in the gut of a cow, the gut of you. They're found everywhere. We just didn't really know what we were looking for. And so if we look on the phylogenetic tree of life, basically this is that first, right here we would say that last universal common ancestor that all life on our planet has. Basically, we had a branch that in this direction that went to form the bacteria, the domain bacteria. Then we had another branch that broke off and formed both the eukaryotes, like you, and then the archaebacteria. What does this mean? Basically, you're more related to an archaebacteria than you are to a bacteria. But always remember that there's horizontal gene transfer. In other words, there's like mitochondria that formed over here, and so there's probably quite a bit of this horizontal transfer back and forth, and so it's
okay. So what he's referring to there is mitochondria, and mitochondria are bacteria-like entities, which there are I don't know how many, hundreds, thousands, usually found in eukaryote cells. So these little entities, they even have their own DNA, mitochondrial DNA, and it's possible. So many people believe that the mitochondria in cells, which convert things into usable energy, are remnants of bacteria of the past. Maybe, or something, right? There's been a whole lot of steps that we've missed. It's noteworthy when he says it's the horizontal gene transfer. I'm not saying he's trying to pull a fast one, but what may actually be happening is that there are phages being used by bacteriophages, viruses of bacteria, which actually can get sent to cells of multicelled organisms, can get through the cell walls of multicelled organisms, and then are received by the mitochondria within those cells. So it's not just a matter of bacteria talking to bacteria and cells talking to cells. There could actually be organisms in the gut biome talking to the mitochondria within cells through cell walls. And this is why the gut barrier, as Jonathan Cooey, gigahomebiological.org, he's been talking about lately, this notion that the wall of the gut is permeable by some viruses is such an important concept, because it's not just because the cell wall, or I should say the lining of the gut is damaged. It's because there are colonies within the gut and the microbiome that are in communication with not just the cells of the body, but actually the remnants of potentially biological bacterial entities of the past that are living within cells. And all of these things are talking to each other. They're all having constant chatter. And how do they chat? They chat through viruses. Viruses is a way to take information, package it up, put an address on it, and send it off, and eventually someone's going to receive it. That's how your cells talk. They talk by messages which are embedded within viruses. That's the role of viruses, folks. That's the role of viruses. And if you take these viral entities and put them in the wrong place or whatever, maybe it's not so good. But that's what it's there for. Anyways, so this is a whole separate phylogenetic kingdom. Archaea, however you wish to say it. There's TED Talks on this.
It's not as simple as that, but know that archaebacteria share more in common with eukaryotes than they do with bacteria.
This is
What are
key.
some characteristics then of archaebacteria? Basically, they're prokaryotic, and that's a term that's lost meaning, but basically it means that they don't have a nucleus and they don't have organelles. So they're not going to have their DNA separated in a nucleus, and they're not going to have things like mitochondria or Golgi apparatus. So they're basically going to just be a cell, And that cell is going to have its genetic material on the inside in like a nucleoid region. So they're going to be that. They're still going to have cytoplasm. They're still going to have ribosomes. They're still going to have a lipid bilayer around the outside. Now, one thing that they also have that's similar to bacteria is that they're going to have a cell wall. So there's going to be a cell wall that goes around the outside of the archaebacteria. In bacteria, this is going to be made up of peptidoglycan, but in archaebacteria, it's not made of peptidoglycan. It's made of a simpler kind of a connecting subunits. We call that an S layer. But the big difference between the two, big difference between eukarya and archaebacteria is going to be found in the membranes. And so remember, a membrane is made up of these things. They're called phospholipids. And so the phospholipids in your membrane are going to look just like this. They have a fatty acid hydrocarbon tail, and then they're going to have a glycerol head with a phosphate on the top. And so if you look at this one, this would be a phospholipid found in you or in bacteria. If we look at the ones found in archaebacteria, well, there's a few things that are going to jump out. First thing that's going to jump out is they're the mirror image. In other words, it's right hand meets left-handed, and so the glycerol head is going to be pointed in the other direction. We're also going to have right here where it connects to the fatty acid tail. In us, we're going to have what's called an ester linkage. So that's going to be right here.
Esther
but in archaebacteria, they're going to have an ether linkage.
I think that's Esther Liederberg.
And then the other thing that they're going to have is these branched hydrocarbons. So those hydrocarbons are actually going to have little branches that come off the side. And sometimes they'll actually form, instead of a bilayer like we have right here, they'll actually form a monolayer. And sometimes there'll actually be chains or rings that form there. And so you might be thinking, well, why is that? Why is their membrane so different? Well, a lot of archaebacteria, remember, can live in these harsh environments. And if you have a monolayer or if you have a more complex hydrocarbon tail, you can deal with bigger temperatures, higher temperatures, and also bigger fluctuations in pH. And so those are some of the characteristics of archaebacteria.
So they found a type of bacteria, an archaebacteria, in an extreme environment. First, it was interesting because they found this enzyme that could handle the high temperatures, the TAQ, I think that's what it's called, the Taq polymerase, or I know I'm mispronouncing that, and this is where I start to sound silly, but I can keep things simple enough. They found something which made PCR possible, but these bacteria, okay, which are now its own family, this archaea family, they have membranes on their cells, which are much closer to the membranes of the cells of your body, of our bodies, of the cells of eukaryotes, of multi-celled organisms. Okay, interesting. Bacteria, because of their cells, their cell walls, they aren't able to deal with the conditions, those extreme conditions, the heat, etc. You would think it would be the other way around, but no. Now we know that we can, through the work of, just look at the work of Max Delbruck, of Hersey, of Joshua Liederberg, Esther Liederberg, and all of the giants, Sidney Brenner, etc. Much of the work that was done for recombinant DNA and advancement of the field of genetics was done by studying bacteria and bacterial viruses or phages. Okay, and they've gotten to the point where they can kind of tweak these things a little bit and maybe even make certain phages or encourage bacteria to make certain phages. But you can't really do that with cells so easy, or eukaryotes. However, if you can take that practice and of things which have been learned through the study of phages, the study of virology relative to bacteria, and apply it to this new family, this archaea, okay? If you can apply it to them, that means that now you have a phage which can actually infect a eukaryotic cell. In other words, if you wanted to create SARS-CoV-2, what you would want to do is you would need something that would make SARS-CoV-2. Well, you can't just, open up a 3D printer and make SARS-CoV-2. What could make it for you? What could manufacture something that could put anything in it that you want and have an outer layer with spikes and everything else that looks like SARS-CoV-2? You would use archaea. That's what you would use to make SARS-CoV-2. If there was such a, that's what you would make. It's amazing that no one talks about this or hasn't talked about it until now. And what you'll see on YouTube is a sudden, there's a flood right now of videos on archaea. A flood. Because the word is getting out. They have been holding back all of this biology. And they've been, encouraging people not to use phage. There's been almost nothing written. I should say there's been a lot of misdirection on archaea. But it's real. It's real, and it involves people that have been so close to the work, the people that we've been studying here, the major historical figures of biology. And I'm not a molecular biologist, so I can't fully appreciate the work that they've done. But I can definitely understand, when people are working together and have intellectual property shared and are involved in espionage together. That I can understand. So if you go to our YouTube and start searching for this, you'll find this guy here, this Anton Petrov, who, always wears the cool T-shirts and has the interesting Russian accent. He has a video, new undeniable proof that our cells evolved from Asgard archaea. So this is the big thing. This is the common ancestor. In archaea, I think it means archaic. I think that's what it's a reference to. And even when talking about them, there's a lot of misdirection. Let's just look at the word archaea viruses. Archaea viruses, okay? Because everyone likes to show pictures of bacteriophages, the little lunar lander things, and we get knocked out with those. But not all viruses of bacteria, and certainly not all viruses of archaea look like that. In fact, if we start looking at images here, let's see here. Here's some types of archaea viruses. Here's another picture here. And let's find one here, because look, there's many different types. Here's one interesting photo here. Here's different viruses of archaea. Look at this one here, Pleolipoviridae. Pleolipoviridae. That's an interesting one. It actually starts to look an awful lot like SARS-CoV-2. Something that has you could put DNA or RNA in it, and it's even going to have your little spikes on it. Under the electron microscope, it's going to look just like SARS-CoV-2. Wow, isn't that just amazing? Isn't it amazing how when you start using the word phage in your dialogue, and you start doing some compound searches, how quickly you get to this? Now, I certainly can't make the assessment or assertion that the only SARS-CoV-2 that ever did exist was something that was synthetic, manufactured using archaea. I can't say that. But what I can say, though, is that with a very simple understanding of the literature, very high level, it is absolutely possible to make SARS-CoV-2 using this. So why has that been one option that no one's mentioned? Instead, you have Andy Kaufman and Tom Throw Pillow Cowan, saying, you can't isolate anything. You can't isolate it. And just throwing confusion around and then, criticizing Robert Malone without ever actually criticizing Robert Malone. They never do. You really want to get into the fact that Robert Malone is perhaps more capable or more likely to know the actual identity of the anthrax letter mailer than anyone else on the planet? Now that's criticism worth putting out there. And that, Robert Malone has been a colleague of the person that went to the FBI in 2007 and said that Bruce Ivins was the most likely person capable of doing that. At that person, Nancy Hagewood, or of course the person they accused was Bruce Ivins, who committed suicide in 2007. And Nancy Hagewood, third most frequent co-author of Robert Malone's postdoc advisor, Murray Gardner. No, they don't bring that stuff up. Instead, it's just this whining about their throw pillows and isolation. Also, never actually mentioning any type of motive. Like, why is any of this stuff happening? Never talking about the Human Genome Project. Never, ever, ever. Never talking about murder. Never saying, let's just stop this criminal childhood vaccine schedule. Never saying it. You know what's happened in the last 24 hours? Another 300 Americans have gotten neurological damage that's going to be considered autism. You know what happened the 24 hours before that? Another 300. As a matter of fact, we are already up to several thousand people, new people who have autism, since RFK Jr. has said that he's going to get to the bottom of it. As a matter of fact, ever since that Brian Hooker and Robert F. Kennedy Jr. were at some scientific Scientology event in 2015 saying they were going to get to the bottom of autism. Let's see. Let's do the math. Ever since then, we have about 35 million Americans. Our youngest 35 million Americans have been subjected to the criminal vaccine schedule, and we have millions of new entrants into a lifelong injury, while they're still trying to figure out maybe it's the adjuvants. So, this term archaea, really important. I found one really great video. It was a TED Talk. Let's see here. Archaea TED Talk. It was this fellow here. The Hidden Role of Archaea in the Gut Microbiome by Duncan Sutherland. So you don't need to send me the link. You don't need to put it in the chat. This stuff's easy to find on YouTube. It's coming out right now. This is 7 months ago. Why is 7 months ago? I'll bet you this guy knew that everything he's going to talk about, I'll bet you this guy knew 5 or 10 years ago. Why now? Why now? In here, it's only a 10-minute video. I'm not going to play it. He talks about, doing analysis of the gut biome and how like 10% or something like that of the gut biome is actually archaea. Or archaea, however you wish to say it. And it's one of the most important ones because it's the one that works that is closest to the wall of the gut itself. And how when a person is born, when a baby is born, their gut, which gets to be used very early on, okay, is not instantly pre-populated with all of these colonies of bacteria. Just, the whole mechanisms by which a newborn has to get energy and food, all right, is different. It transitions when they come into the world. So how do they get the right microbiome? It comes from breast milk. It comes from breastfeeding. And he's going to go into how critical it is in order for the right microbiome to be created in babies to come from breast milk. And without that, your immune system is going to be severely challenged. Not only are you not going to get the right jumpstart that you need, okay, but what microbiome could be in your gut, which could be better than the one that your own mother had? Now, maybe your mom didn't eat great food. It's possible. I'm not saying that, mom is in great health. But at the very least, she's lived long enough to have this new baby. And she's going to have, most likely, the bacterial elements and the archaea elements which are necessary to pre-populate that baby with the microbiome that it needs in its gut. All of this stuff is now coming out. Finally. And I guess this is why so much of the biology of the past just never made sense. It made so little sense, I never felt encouraged to learn about it. But now I think the gloves are off. I would recommend following and supporting Jonathan Cooey. We really need him in this fight. He can take this stuff to this next level where I start to sound silly, okay? Because I can't do the PhD stuff in molecular biology. What I can do, though, I can't believe we're already 55 minutes in. Unbelievable. What I can do, though, is we can go to Vincent Racconello. And where's his new This Week in Virology, okay? This Week in Virology. The bow tie dude, Daniel Griffith. This is the new one. This is from 2 days ago. Clinical update. This is TWIV 1210. So, let's we're not going to listen to this full one. We only need to listen to the first 2 or 3 minutes or so. So, let's just listen to the beginning.
This Week in Virology, the podcast about viruses, the kind that make you sick.
First off, the kind that make you sick. Interesting. Because they're not going to talk about the 99.999999% of viruses which make you live. We're only going to talk about this little eclectic pool of viruses which make you sick.
From Microbe TV, this is TWIV, This Week in Virology, episode 1210, recorded on April 17, 2025. I'm Vincent Racconello.
Wow, and I thought I put people to sleep.
And you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
Look at that bow tie.
It's we're back to anthrax.
Mm-hmm.
Remember all the anthrax stuff?
What?
That was a number of years back.
That was a long time ago, before I knew you.
Yeah,
I remember that. We still didn't figure out who did it. The guy at the FBI who they think did it died, and so that's it.
Committed suicide, right? There was like some question.
Yeah, I think so. But.
Yeah.
Oh boy. Committed suicide. First of all, don't you know this guy's name? It's like, yeah, Kennedy, who that guy with the gun shot him or something. Bruce Ivins, come on, say the name. I know Bruce Ivins was innocent, but even if you think he wasn't, so you don't know his name. Now, it's curious that they're, this little ha. What's funny about it? What's funny about it? I don't understand this. What's funny about someone who committed an act which killed people? Allegedly. That's what they're alleging. What's funny about a disease that kills people? What's funny? I don't understand, I don't think this is funny. I mean, I think it's funny how people misinterpret these things. I think it's funny the lies that they tell. I think it's funny the extent that they're willing to go. But I don't understand this. Why is it funny? Okay. But they're so close to it. First of all, I mean, who said Bruce Ivins could have done it? Who went to the FBI? You know who went to the FBI? A scientist who's been a long time at the University of Oregon, named Nancy Haigwood, Haigwood, who's been a colleague of Robert Malone since 1989. So Robert Malone, Mr. Disinformation, is this close to the anthrax letter mailer, effectively through Nancy Haigwood. Nancy Haigwood is probably the most frequently discussed person in the book Big Shot: AIDS Vaccine, Passion Politics, a Struggle for an AIDS Vaccine, that came out the week after September 11th, 2001, where we talk about John Wolfe, died early COVID, and who else? Gary Rhodes, he died kind of young. And all of these, and this whole collection of people that have worked with Robert Malone in this book. And no, I'm not going to spoil it yet. But of course, Robert Malone working at the Winburn Center, collecting cancer data from the U.S. military. Should there be a large-scale biological terror attack in a major U.S. metropolitan center, and gene therapy gets rolled out on the military after 9/11. All of this stuff it's right under the surface. It's right behind, it's right under the bow tie. It's right under the little picture of the bacteriophage in the back, which encourages us to think that all bacteriophages look like that. That it's impossible for a phage virus, for something that's related to an archaea, which they're only recently starting to talk about, or a bacteriophage to look exactly like SARS-CoV-2. Exactly like SARS-CoV-2. But let's see what they mention next.
Sad times. Well, let's jump right into it. We got a lot to cover. And I'm going to start with a quotation from William Osler. You know, and
What?
Osler, the more you read about Osler, must have been quite a character. He's kind of, some things that we stick with, some other stuff that are kind of questionable. But here's a quotation that I thought was appropriate. The future belongs to science. More and more, she will control the destinies of the nations. Already, she has them in her crucible and on her balances.
Should we say?
Wow. So both of these guys are like eager to have the baby of obviously Dr. Osler. Look how excited they are. You know, I don't know much. I don't know Dr. Osler from a hole in the wall, but I have heard the name coincidentally. Here's another quote I've read of Dr. Osler. Let's see here. What is it? The edifice of medicine reposes entirely upon facts. Truth cannot be elicited from those which have been well and completely observed. Whatever. Sir William Osler, in the book Channels and Ideals. Now, there's a wiki page on Osler. Some consider him to be the godfather of modern medicine. I don't know. There's so many fathers of this and the first persons of that, and I can't keep it straight. I don't know how anyone could. But obviously, this Osler is kind of an important dude. You know, they want to have his baby. Look how excited they are about this guy. And I just happen to have a book called Osler's Web, named after William Osler. Now, it's curious. The subtitle is Inside the Labyrinth of the Chronic Fatigue Syndrome Epidemic, written by Hillary Johnson. And this is a book basically talking about how viruses cause chronic fatigue. Viruses. This is a book extensively talking about who? A Dr. Elaine de Freitas, Wistar Institute, and even her boss, Dr. Hilary Kaprowski. And who wrote the introduction to Judy Mikovits's books? Oh, that's right. It was Hillary Johnson. Because Judy Mikovits, who, as we know from the Mickey Willis videos, is definitely hooked up with Robert Malone, is Elaine de Freitas 2.0. So they're celebrating Osler. Osler's the hero of Hillary Johnson, and probably Mikovits as well, and probably also Robert Malone. But we're supposed to believe that they're combatant and on different sides? Anyways, it's a shorter video. I can't believe it's already an hour. The stuff I'm talking about is right under the surface. It's right there. Not conspiracy. These are the real books. These are the heavy books. These are the books that you start reading them and you don't finish until several weeks later because there's just so much information in them. It's not simply a matter of Oswald, Epstein, and Zionism over and over again in some stupid, pointless loop. These are the real books, and they're making mentions of it because Vincent Racconello himself, if you dig in, was actually said that he was excited about the work of Judy Mikovits back in 2008 and 9. He was excited about XMRV. He was excited about the possibility that vaccines, maybe the biologic element in vaccines has some remnant of a mouse virus. Maybe even they would have gone along with it. Wait 3 or 4 years, to get things fixed up, and then they could roll out gene therapy vaccines. Because it just so happens that if you look at the history, these printed clean gene therapy vaccines, transfections, whatever you want to call them, would have solved all the problems that Elaine de Freitas was bringing up, causing chronic fatigue, and coincidentally Judy Mikovits was bringing up, and coincidentally Robert Malone was bringing up. Because the only reason vaccines don't work is because of the remnants of a mouse virus or contamination or adjuvants or all this other stuff. Every reason under the sun, except the possibility that maybe injecting biologic material under the skin intramuscularly with some attempt, with some desperate desire to reprogram a human immune system is just an absolutely pathetically dumb idea. They just discovered that 10% of, the entire biological diversity of our oceans is something that they don't even know which phylogenetic kingdom it belongs in. We don't even know how many glasses of water to drink. And to think that they actually know how to reprogram a human immune system through intramuscular injection. Come on. Absolutely stupid. Stupid. So, to review, Thomas Brock, PCR. Thomas Brock, definitely read the letters between him and Lederberg. Again, as almost everything that we know about viruses and recombinant DNA, everything goes back to phages. Everything. The person who discovered mRNA, Sidney Brenner, talked extensively about phages. He uses the word liberally in his book. Lederberg liberally used it. Esther Lederberg. Phages. That's what they were all figuring out. That's how they make synthetic viruses. That's how they use CRISPR. CRISPR uses phages. PCR made possible with enzymes that came from archaea. I would recommend searching for ARCHAEA. Learn a little bit about it. Learn about the viruses. It's in one ear and out the other when it comes to all of the specifics of the chemistry. You don't need to know it. You can have a very high-level understanding. Their cell membranes are very compatible with those of most eukaryotes. If you wanted to use a single-cell organism to manufacture some type of synthetic virus which could infect and cause a transfection, you would use something like an archaea to make something that looks just like SARS-CoV-2. You couldn't use a human cell to do it because human cells are not designed, nor can a human cell be manufactured to make SARS-CoV-2. A human cell cannot bud a SARS-CoV-2 virus. It can't happen. It just can't. So where do they come from? Why are people wasting our time still talking about lab leaks? If human cells can't make a SARS-CoV-2 virus, why the hell is anyone talking about it? I'm not saying they don't exist, and the trap is arguing about if they exist or not because you can make one. You just can't have a mammalian cell make one. There's no reason to. That's not the way they work. Anyways, and I think that's enough. So you got some exercises, and but when it comes to really getting all the details, you got to watch Jonathan Cooey's show, Giga Home Biological. He'll get it into that. I need to start getting back to my work sessions. I need to start doing the recordings for the audio book for the rest of the Germs book. Okay, that's what needs to be done. Because it's so strange that this book on biological warfare spends more time talking about the FDA than missiles and rockets and bombs. It's almost as if one of the benefits of a large-scale biological terror attack in a major U.S. metropolitan center would have been to provide the perfect excuse to roll out gene therapy on the United States military, something that would have been potentially highly carcinogenic. Maybe, especially if they didn't use a lot of placebos. So interesting that Mr. Anti-Science Robert Malone, as this falling asleep on the air Vincent Racconello would say, is someone we can't trust. Even though Robert Malone was setting up the data centers there in Windber, where it was capturing that cancer data from Walter Reed as the only center of excellence that Walter Reed had. Walter Reed, if the U.S. military, if the Army has one center of excellence, and if Robert Malone is the guy in our nation setting up that data center, I think that makes him a pretty important person. Especially, as Vincent Racconello may not know, Robert Malone also knew Nancy Hagewood since 1989, third most frequent co-author of his postdoc advisor. And the person that went to the FBI in 2007 and said Bruce Ivins did it 3-4 weeks before Bruce Ivins committed suicide. A name that these idiots don't even know or don't even have the respect to say while they laugh about the anthrax letters. I guess I'm back. All right, guys. Thank you very much. Love you. I got to have a quick dinner right now. And you may notice me on Twitter later, or Twitch later, just doing some recordings. Thank you very much. Hope you had a happy Easter. God bless.